short as possible. So that he is a DVM PhD from UPenn, and after that, he spent a residency and postdoc in Colorado, and he arrived at the U of M 2007. Since then, he is a director of animal cancer care research program, and currently he is a leader of sarcoma twig, and he will be, as you can see in the title, osteosarcoma. So means let's. I'm really looking forward to hear the animal to human things. What kind of things he can tell from the study in in animals in order to understand the human situation. So, podium is yours. Thank you.
Thank you, Masato. Thank you. Can you guys hear me in the back? Okay. And Joel, the mic is transmitting okay? All right, cool. So happy Halloween to everyone. There is a theme to this. So we need to get through some disclosures. You can read them. The important thing is that all of these relationships are managed by the U of M in accordance to its conflict of interest policies. And before I start, I want to give a shout out to Caitlin Callahan, former veterinary student who did a lot of this work with being supervised by Lauren, and then Aaron Sarver and Marissa Maquieto, who really did the bulk of the informatics that I will show you, because I'm definitely not smart enough to do it. So the title, The Ghost in the Machine, the Ghost in the Machine, there's a lot of references, but one is this idea of mind and body existing both alongside and separate from each other. For those of you of a certain age, The Ghost in the Machine was also a very popular album by The Police. And for those of you who don't get this, The Police was actually a rock band from England back in the late 70s and 80s, Sting. Here, when I talk about Ghost in the Machine, I'm going to tell you very little bit about the ghosts that you can't see. So when we think about looking at tumors, there are components of the tumor that are really important, but they're not necessarily obvious. And in osteosarcoma, we have osteocytes, so mature normal bone cells that remain. We have mesenchymal stem cells that undergo differentiation towards myocytes. There are fibroblasts that produce extracellular matrix, and all of these can be very difficult to capture when we actually try to study osteosarcoma as a tissue. And then there are ghosts that are hidden in plain sight. And I am going to be fully transparent and say that I grew up in a T-cell lab, and I'm a T-cell guy, and T-cells are all powerful. And so this is going to be a little bit of a fable and a fairy tale more than it is going to be a Greek tragedy. So the sad thing about T-cells is that neither T-cells nor NK cells can control bone cancer. So much as they try, our superheroes sometimes fail us. And I'm sitting there thinking, like, how can we overcome this? You know, that we're basically blessed with ignorance. So how can we actually figure out what is truly important? So I went to the mirror, and the first thing that I asked is, like, mirror, who's that old guy sitting in front of you? And looks like my dad. But I didn't want the answer to that. So I asked the mirror, mirror, mirror on the wall, who's the smartest of them all? And I don't see that person in the audience, but some of you will recognize that the smartest of them all was Kaylee. And so I asked the mirror, why is Kaylee the smartest of them all? And the mirror said, because she knows that macrophages are it. So Kaylee has been telling you for the last 20 years that you need to study macrophages. And maybe I should have done that sooner, but now we know that macrophages are key mediators of clinical benefit in osteosarcoma, and they really create this, and oh, look who's there, I'm gonna go back. Mirror, mirror on the wall, who's the smartest of them all? There you go. What an entrance, yeah. Vindicated. Yes, vindicated. So, and there is why, Kelly, you're the smartest because you know that the macrophage is key. So I'm so glad that you walked in just at this time. Yeah. So what are we going to talk about today in our fairy tale fable? I'm going to tell you a little bit about what is osteosarcoma. I'm going to tell you about Treatments, I'm going to tell you about immunotherapy for osteosarcoma. And then I'm going to ask you to think about osteosarcoma not as a collection of malignant tumor cells, but really to think of osteosarcoma as a new tissue. So how many of you actually did medical school or clinical training? Okay, a handful, not many. Okay, so neoplasia, for those of you who are into etymology, means new tissue, right? So when we think about neoplastic processes, there really is formation of new tissue. And the pathologists in the previous centuries actually knew better than us. We're so focused on driver genes and mutations that we forget that this is really the generation of a tissue that was not there before. So I want you to go back and think of us as pathologists of osteosarcoma, not as a tumor, but as a new tissue. And then I'm going to spend a little bit of time telling you work that we're doing, uncovering the complexity of osteosarcoma using single cell sequencing, going also into spatial genomics. And then at the end, hopefully it'll become clear why we really need to think about using macrophages as we develop new therapies for osteosarcoma. So the take-home messages are that osteosarcoma and other solid tumors form new tissues that are constrained by developmental programs. So just as you form a liver or a kidney or a heart, when you form these tumors, the developmental constraints of the cells that form the tumors really create that tissue. And so when we think about osteosarcoma in a rabbit or a mouse or a shark, do sharks get osteosarcoma? Kelly says no, why not? They don't have bone, they're cartilaginous tissues. So sharks get chondrosarcomas, but they don't get osteosarcomas. Or humans, these really are not necessarily homologous processes, they are convergent processes. So the way that they start can be very different, but they converge into a very similar appearance because they are constrained by their developmental program. I will also hope to convey that targeted therapies that are intended to suppress the growth of malignant cells in osteosarcoma and probably many other tumors are likely to fail in all but a few patients because the driver genes are not universal. So when we think about targeted therapies, we really have to be thinking about hyper-personalized therapies if we think they're going to work. And that is really difficult to do. And finally, that we might be able to make more strides forward in doing better for patients if we think about altering the composition of the inflammatory environment or the environment in which the tumors live to try to actually inhibit or make the environment hostile to their growth. So we'll start with what is osteosarcoma. So I borrowed this slide from our friends at the Mayo Clinic. And osteosarcoma is the most common primary tumor of bone. It's in humans, it's actually a disease that primarily affects children, adolescents, and young adults. And about 15% of the cases are syndromic, which means that the kids have a predisposing mutation in usually a tumor suppressor gene. And so these kids will develop osteosarcoma, usually along with another panoply of tumors, and they can develop more than one tumor in their lifetime. But 85% are sporadic, so they're kind of the poster child for the bad luck tumor. And it's a very rare tumor. There's only about 1,000 new cases per year in the US. So osteosarcoma of the skeleton is a dreadful inconvenience. But the reason this disease is deadly is because it goes from the bone to the lungs. And all these snowballs that you see, let's see if I can. All the snowballs that you see here in this radiograph are essentially metastatic tumors. So that's the terrible thing. Osteosarcoma also afflicts dogs, and this is the peak of irony with apologies to Harlan Ellison. This is like the story of a boy and his dog, except nobody gets eaten here. And so this is a young patient at Denver's Children's Hospital who is being treated for osteosarcoma, and this is his dog, Saint Bernard, who is actually being treated for osteosarcoma at the same time. And this is courtesy of Steve Withrow, And this is actually one of the dogs in a trial that Steve pioneered to come up with limb salvage therapies that were first applied in dogs and then applied in kids. So this is a pretty old slide. You can tell by the appearance of the building. Denver's Children's doesn't look anything like that anymore. See if I can off. All right. So in dogs, osteosarcoma is also the most common primary tumor of bone. But unlike people where it affects young kids, in dogs, this is a disease of old age. And we see it in big and giant dogs. So if we want to cure canine osteosarcoma, we can just breed tiny little dogs and we will not have that problem. But obviously people like their big dogs. And so this is something that we have to deal with. And it's extremely common. There's actually tens of thousands of cases per year in the US. There's really no definite estimates, but numbers are thrown out somewhere on the low end, 10,000 cases a year, and on the high end, maybe as many as 150,000 cases a year of osteosarcoma in dogs in the US. So in dogs, just like in kids, the tumor of the limb is an inconvenience. It can be managed through use of various different therapies, but the problem is that just like in kids, the tumor goes to the lungs and ultimately is life-threatening. So how do we treat osteosarcoma? So in the last 4 years, we haven't really made much advances in treatment. So if you look at this, these graphs, so this is SEER data. And what you see is treatment for kids that are age 0 to 9 and 10 to 24. And then I cut off the 24 to 40 down here because it doesn't tell you much more. But what you can see is that the introduction of chemotherapy in the 1980s actually made considerable improvements in the outcome from this dark line to this gray line. And then the introduction of neoadjuvant chemotherapy in this population of little kids made slightly more gains. And then in the last decades of the 1900s and then into the 2000s, the gains have really been on learning how to use chemotherapy rather than actually adding new effective drugs. And the gains that we've seen in the littlest kids are more marginal in the older kids and even more marginal in the young adults. And so the two things that really made a difference were first the introduction of chemo and then the introduction of neoadjuvant therapy. And if you look at more recent data from the 2000s, so this is data from the National Cancer Database rather than SEER, and this is looking at increments of whatever 3 to 4 years from 2004 to 2015. Obviously, the 2015 data are not as deep because the paper was published in 22. But you can see that there's really been no difference. So you can pretty much say that from the year 2000 to now, we treat osteosarcoma, and we have not really made any meaningful advances. Importantly, when you look at patients that come in without metastasis, the 5-year survival is not terrible. So 70 to 75% of the kids are alive at 5 years, and about 50% are alive at 10 years. So that actually sounds better than it is. This is still a real problem because you have to think about the fact that these are little kids. So if you are 14 when you're diagnosed and you live 10 years, that means you made it to 24. That's still a lot of years of life lost for these kids. But the ones that present with metastatic disease or the ones that are refractory to the initial therapy have an absolutely horrible outcome. So the 5-year survival is less than 30%, somewhere in the order of 20 to 25%. And the 10-year survival is pretty pitiful, about 10%. So again, metastatic disease is a problem. And when we think about targeted therapies, there's been a lot of trials, and I'm just going to show you data from one paper, but they've really been ineffective. So this is just a reminder, the median survival time for children with metastatic disease is about 30 months. And this is what happens if you take the children with metastatic disease and you give them tyrosine kinase inhibitors. So the two that are most popular now, which are widely used for relapsed or refractory disease, are Rego and Cabo, and big, difficult to pronounce names. But basically, the bottom line is the data here includes various different tumors of bone. So focus on the blue line. Pretty dismal survival for kids with metastatic disease that are treated with rescue with Rego, and not a lot better with Cabo. This actually made the drug actually approved, and this is kind of like the standard of care. But when I've talked to parents of kids that are going through this, they just talk about how dreadful it is. The side effects are pretty horrible, and the outcomes are not that great. So this is definitely not a win. And when we think about dogs, we're in exactly the same place. The introduction of chemotherapy over surgery extended the median survival from about 6 months to about 10 months. And then anything we've tried since hasn't really worked. So this is just data from a paper looking at using standard of care or adding a tyrosine kinase inhibitor. And you can see that the curves are essentially superimposable. Maybe at the end, we make a difference in a handful of dogs that can live a little longer, but not really meaningful. So what about immunotherapy? Immunotherapy has risen as the fourth pillar, and as an immunologist, I'm very excited about this. But T-cell based immunotherapies that have really transformed treatment for several other tumors have also been ineffective in osteosarcoma. So again, a reminder of what the outcome for kids with metastasis and chemotherapy looks like, surgery and chemotherapy. And this is data from one of several trials that used immune checkpoint inhibitors. And so what you can see is that in these patients, virtually all of them had progressive disease. There was one partial response. And the Kaplan-Meier curves down at the bottom, which are kind of small, basically just show that the event-free survival and the overall survival for these kids were not particularly exciting. And this is a slide looking at a trial with patients that had relapse or refractory disease. So they had pre-existing metastasis. Their tumors were HER2 positive, and the treatment used a HER2-directed CAR T cell that is amazingly good at killing osteosarcoma cells in vitro and in animal models. But in actual patients, it didn't do very much. So it extended survival a little bit, but again, we're still talking about maybe a year or 2 years for these kids. So, so far, failures. In dogs, we have seen some glimpses of success with changes in the immune system. So the first observation was that as they were developing limb-sparing treatments, so dog has a tumor usually in the distal radius or ulna, so near the wrist. It's a really good place to actually take out the tumor, put in a graft, and try to keep the limb. But it's a dirty surgery. You have to use a bone from a cadaver to implant the bone in there and let the new bone grow into it. So there were problems with infection. And it turned out that in the initial studies, the dogs that had infections in their surgical site actually did better. Some of them went on to actually be cured. And so this is kind of like a rediscovery of the colitoxin, right? This is from a more recent study where the Veterinary Surgical Society, whatever the surgeons guys call themselves, did a larger study, and they basically saw a small benefit, but in the infinite wisdom of people who are P hackers, they said it was not statistically significant. I'm not sure if it matters if it's statistically significant or not. There does seem to be a little bit of an effect, but it's a very small sample size. So to be determined, but of course, if you are a dog or a member of the dog's family and you decide to do limb spare and that limb gets infected, there's risks of getting vegetative endocarditis, infectious liver problems, septicemia. So you don't necessarily want to actually purposefully infect the lesion. So this is probably not the best idea. But going back to the 1980s, Greg McEwen, who was then at the University of Wisconsin, did a sabbatic at Memorial Sloan Kettering. And he came back from Memorial with a vial of this shmoo that was called LMTP-PE. And LMTP-PE, muranyl tripeptide, is essentially a mycobacterial cell wall extract. So think of LPS as being an E. coli cell wall extract. This is the same thing for mycobacteria. And they actually put this in a clinical trial in dogs, and they saw something remarkable, that for about half of the dogs, or 60% of the dogs, it made a little bit of difference. But 40% of the dogs actually were, by all criteria, they were cured. So these dogs, they did not have metastatic disease when they started. The expected survival time would have been about a year, maybe 20% of them going on to 2 years. And now we have 40% of the dogs going on and living or dying of something else. So this is really exciting. And these studies really provided the impetus to develop LMTP-PE, which is actually now approved in Europe. And this paper came out just a couple months ago, showing that kids that have tumors that express high levels of P-glycoprotein. So these are tumors that are going to take the adriamycin part of the MAP chemotherapy, and they're going to extrude it and make therapy less effective. So these kids are eligible to receive mifemertide or LMTP-PE. And when you separate out the kids that received mifemertide from the ones that didn't, you can see that there is a benefit. And again, if you're into p-hacking, the p-value is, this is an interesting paper, because when they do the overall analysis, the p-value is 0.057. And they say, therefore, it is not statistically significant, which to me is absolutely crazy. So there is a benefit. And in the subset analysis, they get p-values that they like. So we are seeing that LMTP-PE, which is really a macrophage activator, is having a benefit in some of the dogs and some of the kids. There have been other trials in dogs. So there's a really interesting trial where they, this was done at Penn, and they took Listeria as a vector, and they threw in HER2 with the idea that the Listeria would in fact create massive inflammation. The immune system would see HER2 as something foreign, and would respond to HER2. So they treated osteosarcomas where about somewhere between 20 and 60% are supposed to have HER2. In this particular trial, they weren't all confirmed. Some of them were confirmed. But the really interesting thing was that, again, they saw that dogs that received this Listeria HER2 vaccine on top of surgery and chemotherapy had a tail of survivors that accounted for about 40% of the dogs. So this is really exciting. There's a sad and sordid story about this product. It was commercialized. The folks who commercialized it made some changes to the product. There were several different sales involved. The company that ended up taking this in their clinical trial, they saw some Listeria abscesses, so they decided to kill it. There was another product with a separate clinical trial being done, and that study finished in 2025. And it was a bigger study, and unfortunately, it did not replicate. So Listeria HER2 made it into kids as well. I don't think the results of that trial have been reported, so we don't know if this therapy will actually make it, permanently into the clinic. I think, sadly, the therapy for as it comes to dogs is pretty much dead. We did a trial when I was in Colorado, started in Colorado, and we actually brought that trial here. And we were using intratumoral adenovirus fast ligand. And there's a whole story behind it, and I'll tell you a little bit in the next slide about how this works. But what we saw was that there were some dogs that got intratumoral adenovirus fast ligand that had very little inflammation, and some dogs that had a lot of inflammation. And when we looked at T cell infiltration, remember, I'm a T cell guy, right? So we got to go look at T cells. Some of the tumors had virtually no T cells, and some of the tumors had a lot of T cells. And when we looked at apoptosis, again, some of the dogs, some of the cells in the dog's tumors underwent apoptosis, a lot of apoptosis and some had just a little apoptosis. And when we looked at outcomes, so these dogs got intratumoral adenovirus fast ligand, which is kind of an adventure. Then we wait 10 days, we amputate the leg, and we follow up with chemotherapy. And interestingly enough, there's a subset of dogs in the high inflammation that actually have durable responses. And again, we have about, this trial included 64 dogs. This is separated by high inflammation and low inflammation. So the low inflammation dogs do terrible, the high inflammation dogs do better. And again, there's this magic number of about 40% durable responses. So we're very excited about this, but as we study more and more about fast ligand, and I won't take you through this rather complicated drawing other than to say that fast ligand comes in, it blows up fast receptor positive inflammatory cells. If the tumor is resistant, the inflammatory cell death actually creates a soup of inflammation that brings in new macrophages. Those macrophages create benefit. And we think that actually the T cells are latecomers into the party because the macrophages have set the stage for the T cells to work. If the tumors, in fact, are fast receptor positive and are killed by the fast ligand, you shrink the tumor, but you don't get an immune response because there's nothing to respond to. Apoptosis is silent cell death in that case. So we have these signals of immunotherapy, and there's now this information that maybe turning on the macrophage is a good idea. So let's think about how that might work and why. So we want to think about osteosarcoma as a tissue. And I'll use one of our spatial transcriptomic samples to sort of walk you through what we see. So basically, osteosarcoma, if we think conventionally, is a tumor that has very heterogeneous malignant cells. They're very poorly behaved. They're weird. And then we have areas of necrosis and hemorrhage. Okay, and so you can see necrosis here. And then we have these seams of pinkish sort of crystalline stuff, and that is abnormal collagen matrix. So it's abnormal bone matrix, and we call that osteoid. So it is not normal apatite, it is different. And so in this H&E slide, which is a little bit harder to appreciate than a beautiful H&E on a standard glass, this is just a part of where the technology can get better. You can see these lighter pink areas that are osteoid necrosis and then some very cellular areas. And if we ask, for those of you who are really into the wonky part of spatial, here's the data. So you can basically see there's, essentially a lot of sequencing. And this is conventional Visium cytocyst. So every spot that we're looking at is 50 microns, 50. So there's maybe 10 cells, give or take, 8 to 10 cells in every spot. And then there's 100 microns in between the center of the two spots. So there's a fair amount of empty space. But if we ask, where are there transcriptional programs that tell us that there are bone cells or osteoblasts? And so what you can see here with my very clear drawing in blue is that the osteoblasts are basically spread through the cellular area of the tumor, as we would expect. So the cells within that blue area, there are cells that express osteoblast programs. And outside the blue area, we have osteoid necrosis, hemorrhage, stuff like that. So what happens if we ask, where do the macrophages live? So the macrophages live essentially in the medius part of the tumor. There's a lot of macrophages there. What about lymphocytes? Well, there's a handful of lymphocytes, not many. That includes B cells and T cells. They're hanging out sort of randomly all over the place. If we look at this top right, these are the vascular networks. So the tumor is recruiting blood vessels to feed itself. There's a little bit of vascular network here. And then you can't really see them, but there's a handful of fat cells or adipocytes that are probably providing energy to this tumor. And then there's NK cells. It's too bad Jeff is not here, so I can give him a hard time, but there are a lot of NK cells. The tumor is happily growing with these NK cells, and the NK cells really seem to be distributed just in and around the blood vessels. So we're not really sure if they're getting into the tumor or if they're just transiting along the blood vessels. But what happens if we actually look at the compartmentalization? So this basically superimposes the osteoblasts and the macrophages. And so if there were regions that were exclusively osteoblasts, they would be brown. And if there were areas that were exclusively macrophages, they would be blue. But what you can see here is that we get the combination of blue and brown into green. And so basically the macrophages and the osteoblasts are just hanging out together. Every spot that we can see pretty much has osteoblasts and macrophages. They are a big happy family having, you know, Halloween party together. They are not separated. So the macrophages are already there in intimate contact with the tumor. And so the question is, are they helping? Are they hurting? And can we use them? So we decided to actually try to tease out the complexity of these tissues by doing single cell sequencing, trying to understand what the cells are doing. And this is definitely work in progress. So everything that I'm going to show you now is kind of hot off the press. And to do this, we actually took dog osteosarcoma samples. We're dog doctors, so that's the samples that we can get easily. Everything I showed you just prior was thanks to Pari, we're able to get human samples, and we're working with human samples as well, but I'm going to tell you a story about dog samples. So we took samples from dogs that had their leg amputated. We carved out the tumor of that leg, brought it to the lab, disaggregated it into a single cell suspension as best we could, and sent those cells to sequencing. We also took essentially blood samples, and we lysed the red cells, and we sequenced everything that remained. So rather than doing peripheral blood mononuclear cells like a lot of people do, we wanted to keep the neutrophils in there. So we did this using red cell lysis. We had access to several osteosarcoma cell cultures. So these are not established cell lines. These are cultures where we bring the osteosarcoma sample into the lab. We disaggregate it into a single cell suspension, and we put it in a tissue culture dish, and we let the cells essentially hang out for a week. So most of the inflammatory cells float, and we can aspirate those off. Most of the normal cells will essentially die after a few passages, and what we left with is a cell culture that is essentially a pre-cell line. They're enriched for tumor cells. And then we also had access to a spleen sample sort of serendipitously, where we actually had taken that spleen sample, disaggregated it into single cells, and we sequenced that as well. And we basically, you know, took this cell salad, and Aaron took the cell salad and put it on a UMAP plot. And so a UMAP plot, and what you can see here is that the top left corner for each one of these groups is the same UMAP plot. And UMAP is a dimensionality reduction method where you basically compress all of the data into an XY axis. And you essentially get clusters of cells, and the more similar the cells are, the closer they are together. So the distance between two clusters is not particularly informative, but the clustering is. So when you get these clusters, it means that cells are similar. On the left-hand side, so the first four panels on the left are essentially white blood cells. And what you can see is that when we look at the whole panel, we have a distribution of cells that embarks essentially all four quadrants. But when you look at where the blood cells are, all of them are just on one side of the quadrant. So all of this is inflammatory cells, and there's no inflammatory cells on the left side. And on the contrary, if we look at the enriched tumor cell lines, so again, this is the whole picture, and you can see that when we get rid of the leukocytes, the cells are only on the left side. And I won't belabor this right now, but each of these cell lines is actually very unique. So it forms clusters that are not overlapping. Whereas for all of the samples, if you color these by sample, the white blood cells from the different dogs basically overlap. So lymphocytes from one dog are very much like lymphocytes from another, but tumor cells from one dog are very different from tumor cells from another. And if we actually do the same thing for the tumors, what we get is a distribution of both tumor cells and inflammatory cells. So as you would predict, the tumors have macrophages, lymphocytes, et cetera, and they're all in there. So as we've gone through the process, Aaron has been working with Marissa and with Grace to develop pseudobulk approaches, and one specifically that I'll tell you about to actually get a deeper resolution of the data, because when you look at single cells, you might have 200, 400, 800 transcripts. But if you look at its neighbor, exactly the same thing, you might capture 800 transcripts that when you put them together, there's maybe 400 that overlap and 400 separate ones. And so now you've got 1,200. So you can actually, by aggregating the data, you can get a look at greater depth or greater diversity of the genome. We also used a method that Aaron developed that's called, affectionately, we call it GCSS, Gene Cluster Expression Summary Score. And basically this is also a dimensionality reduction method to look at genes that are co-regulated in samples. And then we assessed the relationship between GCSS and outcome in a really wonderfully done clinical trial that was not done by us, it was done by the group at the NCI. And this was a dog clinical trial. So in this clinical trial, dogs are treated with standard of care plus minus sirolimus, which is a rapamycin analog. The idea being that osteosarcomas are highly dependent on mTOR, PI3 kinase mTOR axis. If we would inhibit mTOR, we would change the course of disease. I think the data were very predictable. Sirolimus had wonderful pharmacokinetics, good pharmacodynamics, no clinical benefit. And for those of you who are in transplantation, you will also realize that sirolimus is a really, really good immunosuppressant. So you can use that in the transplant setting. So you might be inhibiting cell growth, but you're also inhibiting the immune system. But the cool thing about this study was that there was RNA-seq and outcome data for 190 dogs in the public domain that we could actually use. So Aaron has been working with this method called MetaCell. It was originally published. He made some tweaks. And basically what Aaron does, and he's in the back so he can answer more questions about this, is he essentially overclusters the cells so that you actually get the most refined version of data from these clusters. And then you can aggregate the most similar cells into these metacell or pseudobulks and do analysis. So this is an example from canine white blood cells. And what you can see is that he gets really nice resolution, which is much cleaner. It's less noisy than single cell data. And so these are neutrophils, T cells, B cells, macrophages, and you can see the unique signals from genes that are highly expressed in neutrophils, genes that are highly expressed in T cells, and so on. So when Aaron applied the MetaCell approach to osteosarcoma cells, here's a UMAP of what the integrated data for multiple osteosarcomas look like. And here is the MetaCell. And one of the things that I hope you can appreciate is that you can actually tell which cells come from different tumors by simply looking at the transcriptional profile. So all these sort of whatever this greenish gray color is, or this orange color is, these essentially are all the tumor cells. So this is one patient, this is one patient. And when you look at the white blood cells in the tumors from these dogs all mix together into their lineages. So all the T cells go together, the macrophages go together. And when you actually do metacell analysis for dogs on the left, mice in the middle, and humans on the right, what you see is really, really similar patterns. So the tumors in the dogs, the tumors in the mice, which are now on the right side, and the tumors in the kids that are on the left side, all basically form these very heterogeneous groups, which no tumor being like any other. And so remember what I told you in the beginning, these are highly heterogeneous. And so if we're thinking about targeting driver mutations or transcriptional programs, it's gonna be one-offs. But the inflammatory response in all of them seems to be quite consistent. There are a few peculiarities that I'll show you as we go through this. So we can actually go back to bulk data and metacell data, and we can basically compare those to try to identify not only these co-regulated transcripts, or what we call these GCSS clusters, but also figure out who is responsible for making these clusters, what are they doing? So when you look at this is something that's really interesting, and it'll become relevant in a little bit of time. When we do bulk RNA sequence analysis on the right, we can actually find that there are some samples that have more, that have greater abundance of transcripts that are associated with G2M transition and DNA damage repair in some samples than in others. And what we can see here is that the signature of G2M genes actually comes from tumors in some of the samples, or some of the tumor cells in the samples, but it also comes from macrophages and from lymphocytes. So what you see here as being present in the tumor, you can actually disaggregate here as to which cells are giving you that signal. So again, we can see tumor cells, macrophages, and lymphocytes. We can also see endothelial cells. So you can see that there are some samples that have higher abundance of endothelial cells than others. And we can find out those endothelial cells right here, so we can figure out who they belong to. Excuse me. Same thing with osteoclasts. So the macrophage-derived cells that chew up bone. And same with immune-specific transcripts. So this again will become really relevant, but you see that there's some samples right here that have really elevated immune transcripts. And we can see that those signatures come from across the realm of the inflammatory cells in the metacell. So one of the really cool things that the metacell can do for us is we can actually divide those immune transcripts into discrete cell types. And so now we can deconvolute the signature into its multiple components. So we can actually see that there are some genes that are essentially boxed here that represent common leukocyte antigens, things like CD45, CD48. And they're present across the board in macrophages, T cells, monocytes, B cells, all of the cells that are there. One of the interesting things about dogs and mice that is absent in kids is that in dogs and mice that don't get neoadjuvant chemotherapy, we see a fair amount of neutrophils, whereas in kids that get neoadjuvant chemotherapy, those neutrophils are gone. Okay, so again, we look at T cell specific transcripts, and now those T cell specific transcripts are purely associated with T cells across dogs, mice, and humans. We look at macrophage specific transcripts anchored by CSF1 receptor. And again, we can see that we see those signatures in dogs, mice, and humans, and the clusters are very, very similar. And again, the neutrophil specific transcripts, we can find those in dogs and in mice, but we do not find those in kids. So why do we think that macrophages are important? So one of the things that we've done before is we have shown that in osteosarcoma, when you look at bulk RNA sequencing, in dogs, samples that have elevated levels of G2M DNA damage repair genes have a worse outcome. So the idea is more proliferation in the tumor, the dogs do worse. Conversely, the dogs that have more immune cells in the tumor do better. So if there's an immune infiltrate in the tumor, maybe the immune system is still, fighting that tumor. And when we do surgery and give chemo, those dogs tend to do better. And so we showed that previously in data sets from our studies, and that replicates nicely here. So what you can see, particularly here, where we actually take the median of samples that have high infiltration of immune cells versus low infiltration of immune cells, you see that separation where more immune is better. And when we look at the cell cycle G2M DNA damage repair, more cell cycle genes are worse. Okay, so this basically replicates what we published almost a decade ago now. So how can we actually use metacell to deconvolute what's going on? So when we go to and ask what is the representation of the non-specific immune cells, we essentially get the same answer as we did in bulk. So this includes all of the immune cells. And again, we see that there's improved outcome from dogs that have more immune infiltrate. And when we look at what's happening with G2M, we essentially get the same response. So the ones that have more proliferation seem to do worse. Except now we can actually tell where these transcripts are coming from. And we can refine that. So we can ask the question is, what happens if we just look at the dogs that have tumors that have more T cells? So now we have exclusively looked at the T cells, and we can point out where the T cells are in the bulk data set, and we can look at outcome. And that benefit with immune pretty much goes away. So the benefit shrinks dramatically. On the other hand, if we look at the macrophages alone, we can now see that the tumors that have greater macrophage infiltration do better. And interestingly, remember in the dogs, we saw neutrophils, and in the mice, we saw neutrophils. And when we talk about neutrophils, they probably represent a spectrum of myeloid cells. So they're not just mature neutrophils. And we see this really dramatic increase. And so this is very consistent with what I showed you with Fas ligand before, that being able to engage a myeloid response with cells that essentially give rise to neutrophils and macrophages improves the outcomes. Now, is this true for humans? So in humans, we don't have neutrophils, but we have macrophages. And what we can see here is that when you look at bulk, we actually do see a much more impressive response, so that more immune cells in the tumors basically means that the kids do better. So which immune cells are we talking about? So we do the same experiment where we take data. We essentially use metacell from single cell data. We look at bulk sequencing in a human trial. And the T cell response is kind of puny, but the macrophage response is quite remarkable. And so what you can see here is that having a lot of macrophages really makes a huge difference in outcome. This is not peculiar to just a couple of trials or subsets. So Aaron has looked at pretty much everything that's available. So there's another dog osteosarcoma single cell data set with outcomes where he can replicate the same thing. And you can replicate these findings of what the cells are doing across numerous data sets for blood. So they're not necessarily related to the tumor, but you can actually use metacell to really refine and deconvolute the signatures we see in bulk. So one conclusion that we're reaching from this is that the presence of an innate immune response that seems to be macrophage driven or macrophage and neutrophil driven is directly related to good outcomes in dog osteosarcomas and in human osteosarcomas. So this really tells us that maybe what we need to be thinking about is strategies that not necessarily just activate T cells like immune checkpoint inhibitors or CAR T cells, but maybe think about things that remodel the microenvironment and turn on macrophages. So we've been doing work with ligand-targeted toxins from Dan Valera's lab. One that's been fairly well developed is EBAT. One that we're starting to work with is DTEGF13. And so these are bacterial toxins that are directed to the microenvironment by paired ligands. There's interesting space for armed oncolytics. So you could think about oncolytics armed with things like CSF1, for example, or GM-CSF, so things that really turn on the macrophage arm. You can think about bikes and trikes, where instead of using anti-CD3 to bring in T cells, you might use antibodies that bring in macrophages into the system and put them close to tumor cells. So there's a lot of opportunities to really think about therapies that might be more effective. So in conclusion, over the last 48 minutes, hopefully I gave you some insights into the fact that osteosarcomas in vertebrates, at least in humans, mice, and dogs, form similar tissues that have similar composition because they're following developmental constraints. So they're not starting in the same place, they're ending in the same place. That when we think about osteosarcoma treatment, the efficacy that we can achieve with cytotoxic therapies is pretty much plateaued. We're about as good as it's gonna get. And tumor-targeted drugs are not really helping that. And that immunotherapies that focus exclusively on T cells have also been not particularly effective. At the end, I showed you that we have some signals or some at least hypothesis generating ideas that osteosarcoma immunotherapies that activate macrophages could lead to durable responses, at least in a subset of patients. And I think if we could actually create these, durable cures with lower toxicity, that would be a huge win. And I think that combination therapies that engage the innate and the adaptive system are likely to be most effective. So I think it is macrophages, but somehow we want the T cells and the NK cells to follow. So in my very unpolitical political statement, I say that in life science and medicine, when we fight together, we win. And some of you might recognize who said that. I won't say it so that no one gets offended. And again, the take-home message is that solid tumors form new tissues, neoplasia, and they're constrained by developmental programs. Targeted therapies that are intended to suppress growth of malignant cells are only going to work in a few patients. And altering the composition of the inflammatory environment of that new tissue has great potential for improving outcomes in osteosarcoma, but probably other tumors as well. So the work happens in the lab. I take responsibility for all the failures. The success is thanks to all the people who work there and do a lot of the experiments and the thinking and the analysis. And in particular, again, I want to recognize Caitlin, who spent 3 years in vet school working on this, and Aaron and Marissa, and of course Lauren, who's in the back, who held Caitlin's hand through a lot of the beginning and supervised her while she was in the lab. And the people who pay the bills, and a picture of my girls, if you guys have any questions.
I have microphones for anyone with questions. Go back here first.
Great talk. Quick question on your neutrophil infiltration association. You mentioned that kind of offhanded that it might just be more myeloid cell infiltration. Are you planning on going back and looking to see if there are any DC subsets that pop out in the survival group?
Yeah, so the answer is yes. I don't know that we have enough data to refine that even further because of the numbers of cells that make up the clusters and the depth of sequencing. But I don't know if Aaron is sitting right next to you, if he wants to add anything to what we can say besides the cells are there and we should be looking at.
So I think couching the term myeloid cells with the idea that they're the most similar to something that has some myeloid transcription is the best way to put it. If you have specific markers for that, we'd be happy to take a look. But they don't look like classic dendritic cells, or they would have popped up. Is the starting point. Whether how far they are along in development, that's an open question.
Great talk, Jaime.
Thank you.
Always love hearing about macrophages. So in your macrophage, and I guess I'm curious if there's a role for osteoclasts at all in this type of tumor, or if they show up in the single cell data.
They do show up in the single cell data, and they're clearly not evenly distributed across tumors. But whether that's because of the bias in capture versus what's really there, we don't know. So as we're going into spatial, we have 8 samples from pediatric osteosarcoma and the high definition spatial incubator right now. So that might tell us a little bit more. So I think right now we can't make a statement about the association of osteoclasts with outcome because it didn't pop out and we haven't looked more carefully. But they are going to be important, and at least to the extent that we can recover them, they're not behaving the same in every tumor. So how the osteoclasts are responding to their own environment, whether they're communicating with the macrophages and the T cells or the tumor cells, and trying to balance which direction they're going to go is an area that's really fertile for more research.
And then I guess my other question, in your single cell data and your macrophage cluster, have you had a chance to dive in a little more closely to look at if there are different phenotypic types of macrophages?
Yeah, so I actually took out a statement that Aaron had put in there that they behave like M1s because, I think what he had concluded, and I will give him credit for this conclusion because it's not necessarily mine, is that the clusters that identify these macrophages that are improving the outcome look more like M1 than M2, and whether that's an intrinsic property in the tumors or whether the chemotherapy is reprogramming to become M1, we don't really know. I hate the idea of M1 and M2 and M0 polarization because the cells are so plastic and they can move back and forth. So we're capturing a snapshot in time, and we don't know what happened 5 minutes ago or will happen 10 minutes later. So I think rather than thinking about strict M1 or M2 polarization, we should really try to think about what the totality of that response is. And it'd be amazing if we could do this dynamically. So we have a trial now in lymphoma where we're being able to collect serial samples. And I don't know if longitudinal analysis will help in lymphoma will help us think about what's going on in other tumors. But I think at least looking at what's happening over the course of days, as opposed to a single time point, will tell us a little bit more about whether those patterns are shifting.
I suspect if you get one, you may be able to find. I suspect that you may be able to find evidence for. If you look for it, you'll find it. Some of these tumors are going to have M2-like activity, tumor-associated macrophages, where some are going to be more traditional, based on the markers and because of the heterogeneity of the disease.
Oh. Okay, I guess this mic died, so I'll stand here. I think the other point to make is that as we think about being we all learn to do reductionist science, right? To look at the genome and then narrow down to the most favorite gene or 3 genes. And I think that a lot of these definitions are based on small numbers of genes, and we should really sort of, as much as we can, try to throw away the reductionist approach and really look at the whole cake. I mean, I know that mechanistically we'll never get funded if we don't do reductionist science, but somewhere along the line, we have to stop thinking about single genes or, handfuls of genes.
I am curious, did you check the gamma delta T lymphocytes populations in the tumor microenvironment? Like, any specifically gamma delta T cells? And the second question is that, like, why dogs get osteosarcoma in the forelimbs and why we are as a human get in the hind limbs? So is there any biology behind those observations? Thank you.
Yeah, so 2 great questions. Gamma delta T cells are present in dogs. Whether their function is similar to gamma deltas in humans is still a question mark. It seems like they do share some traits. We didn't specifically look at them, partly because the mapping of T cell receptors in the genomes outside of human and mouse is not, those are the areas that are essentially kind of a black box. The huge variants and the technologies just haven't caught up. So when we actually recover transcriptional data, a lot of the MHC is either not thoroughly resolved. Well, not either. It is not thoroughly resolved, so we can't actually map to that. When we map, we just don't know where it's going. So it's very, very difficult right now without doing some other things to understand what gamma delta cells look like. There's a really nice single cell paper from UC Davis where they specifically captured cells using antibodies against alpha beta T cell receptors. And I seem to remember that they also sequenced gamma deltas because they have the antibodies and they can enrich them up front. So the answer to your question is we don't know. And I'm not sure that when Aaron did the analysis in the humans that he specifically looked for gamma deltas. Probably not. Yeah.
There aren't very many of them.
Then alpha beta. Yeah. Yeah. And then your second question, the mechanics of motion are really important in how osteosarcomas grow. And so one simple answer, which is probably part of the story, but not the whole story, is that we carry most of our weight on our legs, and dogs carry most of their weight on their forelimbs, so about 60 or 65% of the weight. So when you think about the physical forces on bone and how those physical forces and essentially the mechanical forces translate into cell growth, the mechanoreceptors mediating a lot of cell growth and metastasis, the forelimbs in the dog, the skull and the jaws in the mouse, and the hind limbs in the humans are going to be subject to the greatest physical forces, and that's where we see the tumors. So that may be a teleological explanation, trying to explain why we see something that we don't know, but it sort of fits. So I think that there's mechanical forces that create a more advantageous environment for transformed cells to grow in.
Hi, Jaime. Great presentation. So I have two questions. The first one is when we consider dogs, the osteosarcoma is more older, it means that we suppose that they are exposed to the more longer for the immune pressure. It means having a high immune, immunologically reactive, the tumors there might be reasonable. Then I wonder whether we see the similar percentage of high immune situation in human or not. The second question is, at the beginning, you showed about prognosis factor. One is metastasis. Did you find any factor that related to the metastasis or not? Is that really the character of those cancer cells, or based on the diagnostic timing? So that's the second question.
Yeah, so the first question is, we absolutely see the different inflammatory immune phenotypes in tumors of kids as we do in adults. Whether or not the cells are more or less functional in the pediatric setting, they might be less mature, but they might also be less hyperselected into senescence or exhaustion. It's really hard to say, but having more inflammation is better, whether you're a mouse, a human, or a dog.
Do you have enough number of the patients who shows a high immune reaction in the tumor in pediatrics?
We have enough to actually show better outcomes. So as far as the population analysis, yes. The second question was, is the detection of metastasis biological behavior or timing? And I think it's actually biological behavior, because there are some, you'd never be able to do this experiment in humans, but in dogs, some people will opt to do surgery alone or nothing. And some of those dogs that have palliative, very conservative therapy go on to live for a really long time. So I think that there are some tumors that just won't metastasize. And when we look at the biology of the tumors, we do see differences. So I think that that's part of the story.
Thank you very much. Any other question?
I think we are out of time,
we're out of
so it'd be good if we said thank you one more time to Dr. Modiano.